Now let's talk about neurons. Well, these diagrams are pretty noisy. There are really only three types of neurons, or four if we want to be precise. Sensory input, taking stuff in, association, doing computation, and motor output. And fulfilling these roles that are excitatory, which means positive, do something, inhibitory, which is to prevent excitatory neurons from doing something, and disinhibitory, which accelerates the utility of an inhibitory process of dismounting an inhibition of an excitatory neuron. And there is quite a bit of sophistication in what these, how this process works, which I think we'll cover a little bit, but I'm going to stay away from trying to be overwhelming. I'm just trying to get across a certain set of ideas here, and getting down into the mechanics of all the ways these things can work together is probably not constructive to our general message. Now, in the cortex, about 80% of the neurons are excitatory, which they're the ones that do the learning. Now, inhibitory neurons, well, they don't do the learning. They just regulate or throttle what's going on. Of those 80% that are excitatory, about 80% of them are what's called pyramidal. They have lots of synapses, and 30,000 isn't unusual. About 20% left are called spiky stellate, and they're pyramidal, sort of without the pyramid. I don't think it's valuable for us to get into why the spiky stellate differ from the pyramidal, but it's at this point, it won't help us with our mission. Of the inhibitory neurons, there are at least a dozen different kinds. Cells with inhibitory output connections include this little list: large basket cells, columnar basket cells, double bouquet cells, chandelier cells, and a lot more. There are really good reasons for this, because a lot of these inhibitory neurons form the interaction between the incoming information and the pyramid cells that do the calculation. There are way, way more than that, but that's where I think in our work you're going to see them the most. While there are only three types of neurons, there are many variations on each of them depending on what purpose they are solving, how many inputs they're working with, how much translation is needed to interpret the information, how far the information needs to be transported, and whether it's inhibitory or excitatory. Neurons consist of trees of dendrites that take input, a cell body that carries a voltage, an axon with branches and axon terminals that distribute that voltage when released. Sensory input neurons consist of a cell body or soma, a single pole out of the soma that divides into an input and output axon, with the input axon terminating in dendrites, and the dendrites terminating in sensory cells, and the output axon terminates in axon terminals. They are sensitive to touch, pressure, pain, and within the body they can sense such things as blood pressure. So they are actually sensitive to deformation of the cell body, and that deformation is referred to as mechanical sensitivity. The single pole or unipole neurons serve as relays. The associative neurons are multipolar, with many dendritic branches for inputs, but only one output axon terminating in axon terminals. Motor output neurons are multipolar, with only one output axon terminating in axon terminals. They are interesting largely in that they terminate in a muscle. Dendrite means trees in Greek, and that's what they look like. They serve as the input to the neurons. They receive information from other neurons or the external environment, and they transfer that information to the cell body or soma. They're really numerous. They're short, and they branch in tree-like fashion. They have numerous spines on them to provide a lot more surface area for other neurons to synapse upon, and that word is cool because we mean to say we say there's a synapse, but neurons can synapse to one another. So synapse is the synaptic cleft or the gap between them or the connection, but we can also use it as a verb and say one thing synapses on another. They receive information from other cells at these synapses, and dendrites can grow longer whenever there's activity near their ends. In other words, if they need to create more surface area, they will grow wider or they will grow longer. Dendrites are covered with filaments like leaves on a tree. These filaments are listening or broadcasting, and if they receive information from an axon, they'll develop from a filament into what's called a spine. A spine is a receiving port for a synapse. They're really plastic and they can appear or disappear over just a number of hours. So if your short-term memory and your long-term memory are working or you're focusing on something, we can build connectivity there, and we can, if no longer necessary, we can also release it. So because these dendrites can connect to one another in very complex patterns, there is the general theory that within the same neuron, areas of the dendrite can actually form computations in and of themselves before information gets to the neuron, in other words, the cell body. Now, the soma, or cell body, regulates chemistry and charge. It has channels, pores, or gates of multiple kinds. These kinds include 1 that's open all the time, 2 open for plus and minus conditions, 3 open for they're open by nearby neurotransmitters. In other words, they open in response to neurotransmitters, or they're opened mechanically by mechanical deformation of the cell membrane. They have a means of influencing the soma or cell body. It includes proximity to the axon hillock, the wider the dendrite for information to flow along, the more neurotransmitter that's released, for example, larger synapses, more pores, more reserve of neurotransmitters, and they can move both positive and negative, or pro and con, with the chemicals sodium, potassium, or calcium and chloride. The next part's the axon hillock. Neurons fire by competition between these positive and negative inputs. So positive to excite, negative to inhibit. And the positive increases the voltage and the negative decreases the voltage. Firing is called releasing an action potential into the axon, through the axon, to the axon terminals, and then to the synapses. Firing occurs right here at the axon hillock. Now, the axon hillock consists of sort of like a funnel or a fibrous funnel with hundreds of voltage-regulated channels that function as gates. And firing either occurs or it doesn't. And if it occurs, it releases the action potential, or it doesn't. And there's no such thing as partial firing of a neuron. It's an all or nothing proposition. Neurons fire at their full strength, always. Firing at full strength ensures that the full intensity of that signal is carried down the nerve, down the fiber, and transferred to the next cell. As the neuron fires, the voltage equilibrates, and the process of increasing and decreasing voltage continues once again. Now, I'm not competent to judge the pseudoscience from the science from the mathiness in this field, though I'm comfortable making the following assertions. The information conveyed in a single firing is actually unfathomably complex. Neurons seem to fire randomly, and the reason for this is something that is either unknown or that I'm unable to separate from the chaff for you. Neurons and groups of neurons fire by frequency, amplitude, and phase. In other words, while a neuron can fire as an all or nothing proposition, it can fire by frequency. Groups can fire in numbers, which creates amplitude, and they can fire in phases. So these, then these, then these, then these, and then repeating. So there is an unfathomable amount of information that can be processed by these combinations. The frequency can include rate coding, such as the strength of flexing any given muscle. It's possible that firing patterns also include some sort of encoded information, but everything I've seen so far is somewhere between sketchy and woo-woo. Now, as for axons, an axon means axis in Greek. They provide the output wiring for the neuron. They transfer information to other neurons. They begin at the hillock, which is that swelling at the junction of the axon with the soma. Most are myelinated. Myelination acts as an insulator to help the conduction of action potential. Now, and compared to the dendrites, they can be relatively long, some reaching several feet. They can branch. They can have axon terminals that branch from the end of the axon, and at the end of the axon terminal are bulbs called boutons. Now, the swelling at the terminal bouton is where the neuron synapses with another neuron. And just for some context, recall that our nerves are just axons, and that the longest axon on the human body extends from the tip of the toe up to the neck, and it's something like 15 feet in overall length. As for the boutons, the membrane contains channels for in and out. They contain numerous vesicles or containers that hold these bundles of neurotransmitters. They're sort of like, I think of them as fruit. Think of an orange that's been filled with some other kind of fruit. The space between the terminal bouton and the next cell is known as the synaptic cleft, and it's approximately 20 nanometers thick. Now, bigger boutons, for example, have more surface area for more chemical transfer, and smaller ones have for less. So this is another place where information can be regulated. As for the types of synapse, are we getting complicated enough here? You don't have to remember this. I'm trying to get one idea across to you by making sure that there's no room for woo-woo left. So we're going to continue at this level. The axon can synapse on other cells in various forms. To a dendrite, which is axodendritic, right to an axon, which is called axo-axonal. To another axon terminal or bouton is called axosynaptic. To a soma, axosomatic, releasing only into the extracellular fluid, and yes, they can do that, is called axo-extracellular. Directly into the bloodstream is called axosecretory. And to a muscle, and I forgot the term, in neuromuscular junctions, they synapse directly onto a muscle. Now, dendrites and axon terminals together are interesting. The synaptic potential travels passively along membranes and is described by what's called cable theory. And if you think that sounds mechanical, it is. The cable equation describes how the voltage will change over time and space along a cable. The theory was originally developed for signal decay in transatlantic telegraph cables, but the principle holds for voltage-independent length of membrane like a dendrite. So voltage decays exponentially with increasing distance from the synapse. The extent of the signal decay is governed by the axial resistance, influenced by the dendritic diameter, and the membrane resistance, and the membrane capacitance, and the branching pattern. In other words, if it's really simple and not very long, it doesn't lose too much voltage, but since these things are an all-or-nothing proposition, the more complex and the more surface area they've got to carry, the more dissipation as the charge is carried along it. Now, as for the synapse, when the neuron fires, the synapse also decides. And the synapse decides based on how much neurotransmitter it has in reserve. There's like breathing in and breathing out. The physical size of the synapse and how many spikes have recently occurred. A spike means how many times it's been sent a command to fire. The way this plays out mechanistically is that the neurotransmitter is packed into units called these synaptic vesicles, and which are these spheres, but they have like 5,000 neurotransmitter molecules within them. The vesicles sort of pile up inside the synapse, and a few of them dock on the surface where they form the readily releasable pool. And then when a spike comes in, a small number, an integer number of these vesicles fuse to the synapse membrane and release their neurotransmitters into the synaptic cleft. Now, often this number is actually 0, and this is important. One of the reasons we have a lot of these connections is that the synapses have to keep a supply of neurotransmitters. They can only supply them so quickly, so there's a lot of redundancy here as well as complexity. If the synaptic strength is such that 0.3 vesicles should be released per spike, for example, then this means that most spikes need to release nothing. In other words, not only can somebody run out of something, but they split the job among a bunch of them so that not everyone has to always fire it. Even when they fire, they all don't have to be able to supply something. Several factors determine how many vesicles of neurotransmitter get released on each spike, as again, including none. The size of that synapse, the larger synapse has a larger surface area and more docked vesicles waiting to be released. Synapse size is one of the physical manifestations of what we think of as connection strength. Now, it also depends on how recently other spikes have occurred. Vesicle release is caused by this calcium concentration. Again, I'm going to try to stay away from the charges and the chemistry as much as I can, but the calcium goes up each time a spike occurs, so two spikes close together will generally release more neurotransmitter than two spikes spaced apart. The next variable is the number of vesicles docked in the readily releasable pool. If several spikes in a row have come in, so many vesicles may have been released that there are no docked vesicles left. Time needs to pass for new vesicles to dock. So the takeaway is that the reason axons branch into many axon terminals, boutons, and synapses is largely to guarantee sufficient supply of neurotransmitter release to meet demand. There are also undoubtedly instances of some logical component to these connections, but primarily the boutons can only process so much material at one time and so much over time, so increasing the number or decreasing them can increase or decrease the supply of neurotransmitters. Plasticity or change can occur at dendritic spines. It can occur by dendritic branch growth, by dendritic proximity when they're just close to each other, by dendritic connections when there's a connection there, by chemical composition of the fluid. In other words, there can be information, chemistry in the fluid next to the dendrite, which will cause it to behave differently or grow. By synapsing to the soma, by increasing or decreasing by increase or decrease in soma channels, by connection to the axon directly, by the axon branch terminal or bouton growth, by axon terminal or bouton proximity, by axon terminal and bouton connection. Was that enough variation? Well, that's a lot. Well, we still have, they can go by local, regional, or interregional changes in connectivity information, but most oddly, regional appropriation in response to damage, trauma, stress, or excessive use. So we can actually appropriate, as you probably heard, we can appropriate parts of the brain. Now, this is what's important is we don't seem to be able to grow brand new neurons very easily. We can modify them, the ones we have. They can connect and reconnect in many ways. We can reorganize our brains willy-nilly, honestly, but we can, if necessary, appropriate nearby brain regions, the neurons in nearby brain regions, because everything's connected to everything, if we need to, in case some part of the brain is damaged, but its function is still necessary and the nervous stimuli is still reaching it. Now, I want to caution you that this particular subject of plasticity is polluted with pseudoscience at present, and as far as I know, contrary to the very desirable mythology, we don't really see neurogenesis, and this is why many of these new therapies aren't working other than one or two tiny regions of the brain in the very old parts of the brain. I think those places that might work are those most early susceptible to Alzheimer's, but we'll see what happens in the future. Now, here's a video of an axon in action. Let's watch how it searches for connections. That's enough. You get the idea. Now here's another. That video illustrates the create, read, update, or change, delete, or prune transactions that are familiar to those of us who have worked with databases. And if you combine those create, read, and update transactions with the search function in the first video, you see that the model that we use in software is similar to the one that we use in our wetware, our neurons. But this process is more analogous to how paths form across a college campus, for example. Despite knowing that people take the most efficient routes, architects put down these concrete sidewalks, and people walk efficiently so that new paths form independent of how those sidewalks are laid down. And if something, some incentive is changed or some new construction is built, then paths change and new paths form. Now, roads and rivers evolve by the same means, so we can see this happening not only with humans, but in nature. We take the path of least resistance or most efficiency. So this pattern illustrates the neural economy. Those connections that are used get fed. Those that are not used are sort of starved, basically. And then over time, the memory is consolidated and adapted yet again, over and over again. Because our memories are stored in these physical constructions and connections. So along our journey, we'll see, if we try to understand our memories and our minds, that our brains are out of necessity functioning in a neural economy. As you can see, when we're born, we have the cell bodies and major axons, but we don't have the network of dendrites and axon terminals we see develop over the first 2 years. And while we're sure that neurons develop in utero and are largely extant by age 2, after which they taper off or stop, the debate over ongoing neurogenesis, as I've said, continues with both pro and con arguments, and these studies don't produce clear results because of different means they use to detect young neurons, all of which, at least to my read, are questionable. Now, as we see, neurons develop in density as they learn to carry and make use of more information. But it's a hierarchy, right? Neurons depend on nerves, and neurons on those neurons attached to nerves, and so on. And as we gain an experience, we develop more connections that express greater variations, but we also discover generalizations. So neurons in our brains might mature from the foundation of sense perception to motor skill to higher level reasoning, and hopefully, we eventually reach maturity. This means that there are growth phases to human cognitive as well as physical development. And we can be harmed by poor training of our brains at a given age, by failing to exercise our facilities that are on development at that age in that part of the brain. The most studied examples, vision, of course, as in everything else I've mentioned, but the next is coordination. The next are socialization and problem solving. Lastly, it's higher order reasoning and executive function. So my pet peeve is making boys sit in school and prohibiting them from physical activity and dominance play, which is basically causing them brain damage or developmental disorders every day that they're in school. Which is why, my opinion, we should return to either gender-based schools or at least more physical-based schools with gender-based teachers. So the other is putting children of one grade into the same classroom rather than many grades, boys, girls, and latitudinal races. So let's summarize this bit of neurons we've just covered. I can't figure out if it's too much information or too little information, but we'll summarize it anyway. So what I want to get across to you is that millions of nerve fibers reach the cortex and branch into a region, connecting to many neurons where unfathomable numbers of neurons with unfathomable numbers of connections with unfathomable kinds of connections, despite only switching on and off like a transistor and doing so by a range of positive and negative influences of greater and lesser potential both before and after the firing and networked into series and parallel to one another, producing what's an infinitely complex self-organizing logic. Well, it's complicated. I'm going to try to put this into an analogy that folks with technology experience can understand. In database design, we sometimes sarcastically suggest that we can normalize down to ones and zeros. That means that we can start with a table of only 2 rows, a 1 and a 0, and build the entire database up from there. When programming, some of us are keenly aware that all our human-readable code is compiled down into little more than on and off, or true and false switches, or at least a minimum-sized packet of those switches where, like numbers, some combinations consist of some operations, meaning verbs, some names, proper nouns, some of locations, nouns, some of values, nouns, verbs, adjectives, etc., in a very primitive language and grammar. At the very smallest level, we can recognize those switches, the vocabulary, and the grammar, and we can slowly trace the story one step at a time from this simple language that the computer chips read all the way up to what it is we've written. But when we look at our own neurons, this complexity is such that we recognize the switches we call neurons. We are beginning to understand that a grammar exists, and we can vaguely understand that a story is being told, but the vocabulary and the order it's being told in are so complicated, we can't really, at present, break it down and observe it and trace it step by step. Now, this is largely, in most cases, because everything is done in massive parallel and by so many dimensions of information transfer. The best we can say is we have a fairly good idea today how the brain programs itself with the very basic logic available to neurons, all of which just turn on and off according to some very complex language produced by the dendritic spikes, dendrite, soma, axon, and axon terminals, and the boutons, and the connectivity between the chemistry and the surrounding fluid, the electrical charges within and through the surrounding fluid, and the physical connections that are possible between pretty much every one of those points. And that produces this excitatory and inhibitory stimulation that affects the firing of a neuron and the voltage that reaches the axon terminals and stimulates the bouton to release whatever it can across that cleft. However, this means that while we may understand and describe in general how the brain works, despite functioning in just on or off like our much simpler transistors and computers, the complexity of these logic circuits is something that is, for all intents and purposes, far more infinite. It's probably impossible to compute with any accuracy how much information is transmitted by a single neuron firing in the real world use. The logic of any given circuit is, or how given circuit develops its logic, simply because the number of active dimensions of causality and the massive parallelism and redundancy involved just make it all but impossible. So let's cover the dimensions of neurons. First, we have the environment, which is fluidic and very densely packed and spatial. So there's something that they're in a very densely packed in a fluidic environment full of chemicals that are constantly moving around and serving as resources. They have parts, those dendrites in and out. They have philopedia or filaments, which grow into spines, which make available and can make a synaptic port. They have a soma or cell body, a voltage. They have neurochemicals that alter that voltage up or down. They have parts for in and out of these neurochemicals. They have synapses, axons that have attached to that cell body. And they have an axon hillock, which switches on or off or is an all or nothing when voltage is reached. They have an axon, which is the output, and the axon has branches, terminals, boutons, and synapses. There are 3 types of neurons: input, association, and output. They pass information by excitatory and inhibitory means, with 80% of the cells being excitatory. They transfer information electrically, chemically, and locally. The information content they process is firing, frequency, amplitude, phase, and rate coding. Their connections are to fluid, to the bloodstream, to the axon, to the dendrite, to the soma, and to the terminal. They generate relations, discover and create, read and reinforce, update and change, delete and prune, and they have growth phases, which evolve with us, with our bodies, as sensory, then motor, then cognitive. So that's it for neurons.